and congratulations. So, moving forward with our program, we invite as chair of the Vaccines for 2050, Next Frontiers in Biotechnology, Senior Advisor of Bio-Manguinhos, Akira Homma. He's doctor in sciences for the medicine department in the Federal University in São Paulo, and he has virology degree in as a scholar. He has a degree in veterinary by the Federal University of the State of Rio de Janeiro. He was president of Fiocruz and also the Molecular Institute of Paraná, and he was director of Bio-Manguinhos in 2 terms, and he's a researcher at Fiocruz and also works at Abifina. He was elected as one of the 50 most influent people in the world regarding vaccines. As a speaker on this panel, we invite scientific researcher coordinator Rino Rappuoli from the Biotechnology Siena Foundation. He's a doctor in biological science with the University of Siena. He was an invited researcher at Rockefeller Center and Harvard Medical School in the United States, and he works at GSK Vaccines and Global Health. He's a professor at the Imperial College in London, and he's a molecular biology professor at the University of Siena. He's among the medical science leaders, and I pass the floor to Mr. Akira Homma. I rather speak on my feet. Thank you very much for the presentation. I will pass the floor of Rino Rappuoli, and the great challenge for me.
But surely Dr. Rino Rappuoli will, with his experience and his great deal of expertise in the work he's been doing, he has developed various vaccines, reverse engineering, it's a group B vaccine for meningitis, among other things. Dr. Rappuoli is an old acquaintance of ours, and I don't want to take up time. I'm going to draw on this opportunity to learn even more and to see how this challenge will be overcome. By 2050, Dr. Rino, please, you have the floor.
Well, thank you, Dr. Akira. It's always a pleasure to be here and talk to you. And I really want to congratulate Akira for organizing this symposium, but also for being the voice of vaccines for Brazil, at least as far as I remember. And it's been always open for innovation, for collaboration, and for science. So really, it's really fantastic. Even the session that we just had about the G20 is very interesting for me because, as you know, Italy right now is organizing the G7. And most of the themes you are talking about resonate also in the G7. So I think it will be very important that we coordinate and we basically give to the politicians, to the policymakers, exactly the same messages. And I saw a lot of things which are aligned. Having said that, today I'm going to talk about vaccines. And when I talk about vaccines, I will always try to start with this slide. that tries to summarize the technologies that have been used to make vaccines. Well, most of vaccines for 2 centuries actually have been done. Let me see. Okay. Have been done with this technology down here, which is basically what I call empiric vaccinology. Means that at the beginning to make vaccines, you will take a pathogen, a virus, a bacteria, and a parasite. You will kill it and inject it, or attenuate it and inject it. That was the empirical vaccinology from Jenner 1796 to Maurice Hillman 1960s. And I think when Akira and I started to make vaccines, those were the only tools that we had. Up to the end of 1970s, probably. Then something happened. 1980, we started having the first revolution in technology, molecular biology, recombinant DNA, all these kind of things. And then we got the vaccines, the glycoconjugations. And then we got the genomics. And today, we have an explosion of new technologies that we are using. So basically, Akira and I had the fortune basically to live through an incredible period where basically many things in the 1970s were impossible became possible. And actually, today, most of you that are young people don't believe that those things were not possible at that time. And it's amazing what happened. So and obviously, every wave of this technology, we conquer new diseases. And that was the beauty of doing that. Now, what I'm going to do today, I'm going to go through the key steps. The key steps that basically transformed the field of vaccinology. I will not mention all of them, I'll mention the most important. And the most important in my mind are reverse vaccinology, structural vaccinology, and synthetic biology. And we'll walk through these while we go to the end and we look at the future. So, reverse vaccinology is the one that basically started mid-1990s. And the reason was that we, in our laboratory, but many other groups worldwide, including here in Brazil, were working trying to make a vaccine for meningococcus B. And we tested all the technologies we had. We had tested the genetic approach, we had tested the biochemical approach, the purification of proteins, killing bacteria, making vesicles from the bacteria. All the technology that we had didn't work. And then was a moment where basically the vaccine was impossible. It was impossible, at least with the technology that we had. At some point, Craig Venter at that period basically described a new technology, which was basically sequencing a genome of a bacterium. That was clearly a new revolutionary technology. And we said, well, if it's possible to sequence the genome, maybe if we get the sequence of the bacterium, we will find in the genome those things to make a vaccine which are impossible with the technologies of today. That was the thinking, and at that time, I went to visit Craig Venter in Rockville in the United States, and we made an alliance. The alliance was basically Craig Venter was the man that was the best to sequence genomics at that time. Richard Moxon in Oxford was an expert geneticist, and us who were good at making vaccines. And basically the idea was Craig Venter makes the genome, we interpret the genome, we try to see whether we can make a vaccine. And we did. I asked the computer which were the genes that possibly could code for a vaccine molecule. The computer said 600, and then we tried to express all the 600. We succeeded in 350. We found 91 which actually were good potential vaccine candidates, 28 which were very good vaccine candidates, and 3 that we selected at the end, and eventually we made a vaccine. And that was a vaccine was simply impossible with the technology we had before. Now, that was the first time genomics were used to discover something we couldn't do before. And now, this was 97, 98, the Stone Age of genomics. Took to Greg Venter 14 months to make one genome. Today, you know, we make hundreds, thousands, how many you want to do in one day. Things have advanced, but that was the first time that, if you like, genomics, big data, or whatever you want to call it today, were used to make something which was impossible before. The second thing I want to mention is structural vaccinology. Now, genomics, when I started, basically to sequence 200 bases of DNA, it took 6 months. When I started making the structure of a protein, was taking a year, 2 years. Many scientists would spend the entire life in the structure of one protein. But then 2007, 8 became clear that it was becoming easier and easier to make structure of proteins, easier to crystallize proteins, easier to basically cryo-electron microscopy started to come, and became clear that at some point it was going to be possible to have high throughput structure of proteins. And where the structural proteins made the difference was respiratory syncytial virus. Now, I'm sure you know about respiratory syncytial virus. It's a virus like many others. You see there as a typical virus with proteins on the surface that we call spike, or we call F protein in the case of this. And so, and it's responsible for a lot of mortality of newborns in low-income countries. And most of the hospitalization in medium and high-income countries of children in the first 6 months of life are due to respiratory syncytial virus. It's so important in the 1960s, they wanted to make a vaccine. They made one. 1967, it tested, was a disaster, didn't work. And since then, many times they tried to remake a vaccine. 1970, 80s, 90s, 2000. Last failure was 2019, so yes, yesterday. And we failed for more than 50 years. And the question was, why? And why the science didn't help us? And the main reason was that we had no idea what we were doing. And I'll show you why. Basically, you see in this slide is the structure of the protein, which is F protein, which is the surface of the virus. That's, if you have antibodies to this protein, you are protected. And people try to make, through vaccines, antibody to this protein. This protein was present in the killed virus, in the live attenuated virus, who have been tested in clinical trials. But nobody knew why it was not working. Eventually, a group at the NIH, the group of Barney Graham and Jason McClellan was a young science doing that, got an antibody that will bind the protein, and that could do the crystal structure. And the crystal structure showed that basically the proteins, which is a trimer, has this conformation when bound to the antibody. But in the meantime, our lab had done the structure of the protein and had this conformation, same protein, same amino acid sequence. What was the problem? The problem was that the protein on the surface of the virus is like this, it's called prefusion, but it's very unstable. And if you make a vaccine with this protein, you go and get a coffee, you come back, the protein is gone, because it's very unstable, it's moved from this conformation to this conformation. The problem is that this protein is a good vaccine, this is not a good vaccine. So for 50 years, we have been working with the wrong protein. Well, the protein is the same, but the wrong structure of the protein. And now, when the two structures became known in 2013, then the question was, how do we stabilize the protein in this structure? And down here you see, this is a trimer. Down here you see a monomer. So this is a structure of the protein that you want to do as a vaccine. This is the protein that you don't want. How do you make it? Well, look at these two red amino acids. They're very far apart in the protein you don't want. Look how close they are in this structure. They're one next to the other. So Jason McClelland did change the two amino acids into two cysteines. Two cysteines make a disulfide bridge. Done. That protein cannot move anymore. That's the vaccine. 50 years of failures. Now, I'll tell you, my grandchild, 6 years old, if I give that thing, he's going to say, well, this is enough. You put the glue there, it's going to work. It's easy now. But for 50 years, it was impossible. And the reason it was impossible, we had no idea about the science. And now that we know the molecular structure, it becomes easy. So that, the consequence of that is that was 2013. In 2023, we basically got 2 vaccines approved for the elderly by the FDA, one from pregnant women, and one monoclonal antibody recognized that thing. Now things are easy, just because we have technology, we have the knowledge. So the next thing came in Jason McClelland, the person who made, engineered the protein RSV. In 2017 was working on something that nobody could care about, which was MERS, which is the coronavirus of camels in Saudi Arabia. Nobody was interested, but he looked at the protein on the surface of the virus. He basically found that when you look at the microscope, some proteins were like the bad protein of RSV, and some proteins had the right structure. So he said, nobody cares, but it looks to me that if we one day want to make a vaccine for a coronavirus, we need to stabilize this protein. And then this time he looked at these 2 alpha helices, which are connected with this bridge, basically. And when they move to the post-fusion, the wrong protein, they move from being like this to being like this. And so basically he said, how do I make this impossible? Well, it's easy. Just put a proline instead of this asparagine here, and then that blocks the thing that was done. So nobody was interested in this in 2017, but in 2020 when COVID came, basically this paper that nobody wanted in 2017 was the key paper that made possible all the vaccines for COVID. Okay, that's about structural vaccinology, but you see how important is the structure. Now, I'm going to tell you about how we move from reverse vaccinology also to synthetic biology and what I call digital vaccinology. And to do that, I want to bring you back in 2009. 2009, you remember we had another pandemic, the swine flu pandemic, was not so, basically, was not so severe, so very easy to forget. But for people like us, that we were working in companies making vaccines, was a very important moment. And the moment went like this. Day 0, basically, the virus was found in Mexico and then southern United States. They all called us at the BARDA, the NIH, said we need a vaccine. Now, we knew how to make influenza vaccines, so we started immediately. But to make a vaccine, at that time, what you need to make a vaccine, you need a virus. So the virus, fortunately, was in United States. And, to make an influenza virus vaccine, you need to take the virus, adapt to growing eggs, and then you make a thing. That takes a couple of months. So the virus went to CDC in the United States. The CDC gave the virus back to us manufacturers. We started to make vaccines working days and nights. And we made vaccines. And we were actually faster in making vaccines than we have been with COVID, just because we had the manufacturing size, the eggs ready. So we made a vaccine, and in 5-6 months, we started to have enough vaccines, and then we make hundreds of millions of doses. However, when we got enough doses of vaccine, it was very sad because the virus had gone had its peak much earlier than we had the vaccine. And basically, so the vaccine was too late. So we got a vaccine, we made it, nobody, at the beginning, we were interacting with the Ministry of Health worldwide. The day before, they were saying, we need your vaccines at all costs, and 1 month later, we don't need your vaccine anymore. That was so basically very disappointing. The message was very clear. We need to go faster. You need to go faster. So basically, this was 2010, 2011, 2012. Every person working in influenza or in vaccines had in mind we need to go faster. The next thing that happened was the Easter Day of 2013. 2013 Easter Day, the Chinese Center for Disease Control were basically put on the website the sequence of influenza virus, H7N9, that already killed 3 people in China, had all the properties to, was an avian influenza, was all the property to become pandemic. At that time, we were very scared about potential influenza pandemic. And basically, the alarm sound and basically said, we need to make a vaccine, like in 2009. The problem was that this time, the virus was in China. And took several months before the virus could get out of China. We could start to adapt to eggs, all this kind of thing. So that was the moment I was still collaborating with Craig Venter, where we basically decided to take a totally different approach. And the approach was the following. So on the Sunday, basically the Chinese put on the internet the sequence of the virus. Monday morning, the lab of Craig Venter, this time was in San Diego, California, downloaded the sequence and started to make 2 synthetic genes, the magglutinate and the neuraminidase. Monday night, put those 2 genes into express mail. Tuesday morning, arrived in Boston, where at that time I was working for Novartis. We had our labs for vaccine research there. And basically, we took those synthetic genes. Tuesday morning, we started to make 2 vaccines. One, we used reverse genetics to make a virus to be used to make a conventional vaccine. The other thing, we made RNA to make, and by Saturday, the same week, we had both vaccines. We had the virus to make to make a conventional vaccine, growing in eggs or growing in cell culture, and we had an RNA vaccine ready to go. Well, that was interesting. We had the vaccine, we had never seen the virus. And then we immediately came, this is a slide that Phil Dominster made at that time, and said, but why are you so stupid? We're shipping around viruses from one country to another through custom difficult virus. It's so easy. We only need to ship the sequences. And that was the moment I thought we changed from what I call analog vaccinology to digital vaccinology. Why this thing? Because, I mean, before, to make a vaccine, you needed a virus, you needed a parasite, you needed a bacterium. Now, all you needed was a sequence. And then the sequence of the genome goes to a computer designs, can design your vaccine. The information goes to a robot, and maybe you can have a vaccine made by a robot without doing anything else. That's more or less not too difficult. Now, a little bit of science fiction. 2050, you know we're going to have a colony on Mars. But at least that's what they say. And so basically now you have a pandemic or an emergency, and now at that time we'll have solved all the problems of equity and making vaccines for every piece of the world, because the sequence will go to the internet, the computer will design a vaccine, and the computer will send information to many robotic stations which will be present in every single part of the world, and those robotic stations will make a vaccine. So it will not be a vaccine for US and one for Africa, Asia, Latam. Every place will have a robotic station that's going to make a vaccine. But we also have a colony in Mars. Now, a colony in Mars, if you want to send a vaccine there, with a shuttle, it's going to take 9 months. But if you actually send the sequence by information, it's going to take 2 days. So, and you say, well, this is science fiction. It's true. But to me, the science fiction is more the colony in Mars than the rest. It's not impossible. It's coming. So that's science fiction. Back to reality. When in January 10, 2020, still the Chinese Center of Disease Control, George Gao, the man who talked yesterday to us, put on the internet information of COVID, of the SARS-CoV-2 virus. Basically, this time there were more or less 350 labs worldwide that were able to do what we had done in 2013, download the sequence and make a synthetic gene. And then the synthetic gene was used to make RNA vaccines, all synthetic. So basically, Moderna was able to do in clinical trials in 66 days. Others took the synthetic gene, put on an adenovector or other viral vectors, took a little bit more, but Oxford in 3 months was in clinical trials. Others put the synthetic genes into mammalian cells like CHO, insect cells, or tobacco cells, made the protein, purified the protein, combined with an adjuvant, and then went into clinical trials. But everything started from the synthetic gene without, at the beginning, knowing the virus. And I'll not go into what happened later about the COVID vaccines. Everybody knows more than I do about that. But I only want to make some reflection about what happened. Basically, the COVID vaccines were basically, the first one was against Wuhan. And people say, well, they don't work. We keep being infected. What's going on? What's wrong? So basically, I've gone back to 1970 test book of immunology and took this red line, which is the shape of the antibody response. And basically says that when you immunize 1 or 2 doses very close by, you get a primary immune response. It goes up and comes down. But then if 5, 6 months later, or even 20 years later, you give a boost or another infection, you have a secondary response. Now the antibody goes very high, and they come down, but they never come down to the levels before. So down here is basically the number of antibody units that you need to neutralize the original virus, Wuhan, 12. I mean, if the virus are not changed, we would basically have a vaccine that will last lifelong. But then we got many variants. Here I put Delta. Delta, you need 76 units now to neutralize it. So the secondary immunization was still good, the boost, the third dose, but the primary immunization was not good. And then comes Omicron. The initial Omicron required 1,100 units. And the new variants require 7,000, 8,000. So basically, now we are in this condition where basically the new variants, when you give a vaccine, you boost a little bit, but then immunity goes down. So vaccination protects from infection for a few months, but then you become susceptible again. Fortunately, the all vaccines that we have protect very well from severe disease. So that basically we are pretty happy about that. So that's the thing. I mentioned this 2 days ago. I just want to remember that not only technology was important to go fast, it was also the decision to invest by the public sector, basically, that allowed us, instead of doing the phase 1, phase 2, phase 3 in sequence, which will take 10, 20 years to make a vaccine, they allowed us to do everything in parallel, and the vaccines were made in 10 months. started to go a little bit towards the future, I want to talk to you about another thing, cancer. I mean, people wanted to make cancer vaccines since 1920s, and there have been thousands of failures so far. Now, the first one that really, to me, seems to go in the right direction is this one, because it's a trial where you have a control. So basically, and this is a collaboration between Moderna and Merck. And basically what is about melanoma, stage 3 people, or 4. They took 57 patients that the cancer had been removed surgically. And then they divided into groups. 50% got the classical therapy that you give to melanoma, which is checkpoint inhibitors, antibody against PD-1. And we already know that these things are kind of miracle for cancer. And so the other 50%, they gave not only the inhibitor, but they gave a personalized vaccine. How was made this personalized vaccine? Basically, they took the sequence, the genomic sequence of the tumor, they took the genomic sequence of the healthy tissue, they looked at the differences, and then through artificial intelligence predicted which were the epitopes which were different between the cancer and the things, and selected 34 differences, and made the vaccine like a sausage, basically, RNA vaccine with 34 different epitopes derived from the sequence of the genome of the tumor. And then the results, which are amazing, basically, the ones that had the cancer vaccine had reduced the risk of recurrences or death by 65%, compared to the other ones, which were already doing well because they were receiving the PD-1 inhibitor. So basically, it's not, I mean, we need to wait. They're doing phase 3 right now, so we need to wait. But this is something, it's the first time that I see something, I think cancer vaccines are coming. So what about the vaccine of the future? I mean, you're seeing all the technologies that accumulated. So basically, looking at vaccine of the future, basically, the design of the vaccines, the discovery, the design, the vaccines, is all going to be done by artificial intelligence, genomics, reverse vaccinology, structural vaccinology, all these kind of things. And this is not the future. This is already now. It's happening right now. We don't put anymore a molecule, a native molecule, into the vaccine development, because we know that when we isolate molecules, but they are unstable, they can be proteolytic cleavages. Now we design through artificial intelligence how do you make mutations to stabilize the molecule, basically to make sure that the epitope we want are really exposed in the right way. So basically, the design is already artificial intelligence. What about manufacturing? Well, fermentation, filling, they're all things that can be done more and more through robotics. And we know when you have a robotics that basically performs many times the same operation, then artificial intelligence is, I mean, machine learning, whatever you want to call, comes in and basically makes things easier, more reproducible, less failures. So that part is coming, and there are all these ways they call digital twin. Basically, you do the process in silico and the process in real life, and then you compare and improve each other continuously. So basically, that is going to happen. Where there is still discussion is clinical trials, regulatory things, because if you talk to the optimists, basically, they say, oh, clinical trials will be revolutionized. Patient recruitment is going to change. The trial design is going to change. Data harmonization is going to change. Everything is going to change. If you talk to others, they say, especially regulatory agencies, they say, well, we are pretty happy about the way we organize trials, the statistics which is behind. We don't want an artificial intelligence that we don't know what kind of bias is going to introduce to tell us what the result is. We want to see the result. So, I mean, I'm not saying it's not going to happen. It's going to have an impact there, but we need to be careful because regulatory agencies, also patients, also me, I would like to see the real data. I mean, I would not like to see simulated data, whether something is going to work or not. This is pretty open. It's going to be a discussion. Actually, at the end of June in Siena, we are putting together like 100 people to discuss how artificial intelligence is going to impact vaccines. What about targets with all these technologies? So here I divided all the possible vaccines from very easy in green, slightly more difficult in yellow, more difficult but possible in brown, and where our limits are today, which I would say technically impossible in red. And so I will not go through the green. That are the vaccines most of the vaccines that we have. The one in yellow are vaccines that we can make, but basically in order to make those vaccines, we need to vaccinate people already immunized, so it's not so easy like mice where. And then the one in brown are the ones that were impossible up to a few years ago. For instance, herpes or zoster. Zoster is a good example. I mean, we are infected by zoster, by varicella virus at birth, and then we keep the virus for entire life. And most of the life, our immune system is able to control the virus. But then when you get old or when you get immunodeficiency, the virus comes back and you get zoster. Now, we have a very good vaccines for zoster today, and we know that we can make this. So herpes, Epstein-Barr virus, all these kind of things, I think is going to come. Where we don't have a solution yet are basically the one in red. Because in this case, for these ones, the immune system was able to control for most of the life, and the disease comes when the immune system ages or you get immunodeficiency. These ones are when the immune system has been defeated, has never been able to control. HIV, that's the case, not control. Human chronic papillomavirus, no way to attract it with vaccines. Chronic hepatitis C, hepatitis B, none of the vaccines are working with those. And cancer, I put there cancer, because so far it's been very difficult. I gave you an example, but still, even in cancer, the immune system has been defeated, and really, we don't know yet how we're going to do that. I mean, are these things impossible? I don't think they are. I think you have seen the first move of them in cancer, so basically I think these things are going to come. So I want to conclude this vaccine part. Basically, I told you the technologies we have, what we can do, the things that we can approach, the things we cannot approach, which are not that many anymore. In fact, today we have the technologies to make most of vaccines, with except the ones I showed you in red. So the question is, which are the challenges? Or do we have challenges in vaccine? Well, we do. I mean, it became very clear during the pandemic that we would like to have vaccines that last longer. So duration of the immune system, how do we do? I mean, frankly, the science has no idea how to make a vaccine last longer. Then we would like to make vaccines that are going to have a CD8 killing cancer, killing things. I mean, still, we don't know very much about it. Mucosal vaccines. We would like basically to block the infections, the portal of entry. So oral vaccines, intranasal vaccines that will basically not only prevent disease, but also prevent infection. We don't have those yet. We would like to have 100 days vaccines to face pandemics. We don't have them yet. I mean, the last one was fast, but it took 350 days. So the CEPI, the NIH have put this challenge of basically making the next vaccine for pandemics in 100 days. We are not there yet. And obviously, all these things have to do how we're going to do a regulatory approach, put all these things into the hands of regulatory agencies so they can do things. I think with this, I'll probably stop there, and hopefully I gave you an overview of where the vaccines are going. And, well, we'll see each other probably in a few years and see how many of these things are going to happen.
Let's take questions from the floor. Yes, it's open for questions. Please. Yes, please. Take a phone, microphone.
Okay, Dr. Rappuoli, first of all, thank you by this excellent presentation. I have a polemic question for you about bacterial vaccines. We know that it's very difficult to have a good vaccine against Staphylococcus and E. coli. Mainly because this bacteria belongs to our microbiota, so our host immune system don't know this bacteria as a true enemy. What your opinion of how we can do to overcome this situation?
Well, E. coli and Staph are two bacteria which are actually quite difficult. I mean, the problem is that in the case of Staph, there have been several failures. And the problem is that we try to make vaccines using one or two molecules, not necessarily the best ones, or what we thought were the best ones. And the approach was basically to try to make a very high-profile target, which will be very profitable but high risk. So we went for high-risk projects, so the trials failed. I think it's one of those areas where we wanted to jump the science to get to basically, let's see whether it works. It didn't, and now it's people are scared to go back and try to do it again. But I think what we need is basically a kind of systematic approach, do the science well. I think a Staph vaccine is possible, but the Staph has got so many immune evasion molecules that we need to know exactly what we do when we do that. And nobody has been able to approach this from systematically. So we tried to go fast and we failed. For E. coli, I think there are two approaches. Either you go with the LPS, and then that's one approach that's being tested, which is unique, or you go for proteins which are present in the pathogenic E. coli and absent in the commensal E. coli. There are not that many, but there are several, and I think, again, it's possible. It just, for most of these things, it's just a question of investment. You know, when we talk about vaccines, we always talk about the fact that, say, if you take a vaccine which is successful, from the beginning to the end, it's going to cost $1 billion, at least. But in fact, every vaccine is going to cost 30, 40 billion because you have to count all the failures, all the different projects, all the different things. So basically, if we want a vaccine for Staph, the public sector or someone has to put down not $1 billion, because the $1 billion is going to work only to develop a vaccine that we know is going to work. You need to put down research for failures and for doing proper research. So that's what society needs to do, basically. You have many projects, many of those will going to fail until you get the one that's going to work. Yeah.
Thank you, Dr. Rino. One more question? Yes, please. Synthetic question, okay? So that
I just want to hear from you. Well, your talk was very inspiring. Your talk was very inspiring. I just want to ask you one thing very simple about IP problems that we raise when we have all this vaccine made by first by one company. They will protect, and then the others will not be able to do the same using the same technology or something like this. So for a global public health response, we will face this kind of problem. I would like to hear from you how to overcome this.
So you're talking about intellectual property. Well, intellectual property is necessary. I've been in vaccine companies for 40 years, and unless you have intellectual property on the vaccine where you invest, nobody's going to give you money to invest. So basically, that's essential. The question is, when the intellectual property becomes a problem for equity, for instance, like happened during the pandemic. But if you look during the pandemic, Moderna, Pfizer, they said, you can use our technology. I mean, if you use it, we'll not enforce any intellectual property for the pandemic, obviously, not for the other commercial thing. So in that case, it was not really a problem. All the IP was available to anybody that wanted to use it. So I think, and now I think in the pandemic treaty, which is being discussed and discussed and discussed, hopefully signed, a lot of these things should be part of that.
Yeah. Thank you. Thank you. Another question, the last question from the floor.
Yes, which one is the best vector in your opinion, vaccine vector? And a vaccine has to have high immunogenicity, again, and make a lot of antibodies. But how does that correlate with the CD8 T cell response in some cases?
Well, if I understand correctly, the question is how do we induce CD8 T cells, which are very important, especially in clear viral infection of cancer or other things. And in fact, CD8 was one of the challenges for future vaccinology because we know that protein-based vaccines don't induce CD8, even if you use adjuvants. We know that adeno or viral vectors and RNA induce a little bit of CD8, but not too much. So it's one of those areas where I think we don't have yet the technologies to be, to be able to produce systemically, consistently a CD8 like we will do for antibodies, for instance. So it's one of the areas, one of the challenges for the future.
Well, thank you. I have also one question. Yes, I was waiting, but see, I am sure the advancement of science and technology is so fast, and we will have new technology, new vaccine, but the problem, we have also another problem, that the people must have information, must have very transparent information on the purpose, on the importance, on the prevention, on the everything, and take the vaccine. You know, I think this is what we always say. The ultimate mission of vaccine is to eliminate the agent. Polio is almost there, but already for more than 20 years it's almost there. You know, how to get these people in 2 countries taking the vaccine and not using anymore the oral attenuated vaccine. But we know that, but it's very difficult to get this accomplishment, no? And we know that there are many other diseases that we can eliminate if we have high coverage. We can eliminate for example, HPV. No, Australia showed there, if you vaccinate properly, a good strategy, a correct strategy and so on, you eliminate diseases. So we can do that, but there are many problems with HPV, vaccination coverage, and also the disease there. I think hepatitis B, we can eliminate it. If we have good vaccine, and if we have high coverage, well, what do you think? We can eliminate diseases? This is one question. Second question is, there will be one shot for all diseases?
I knew that the most difficult questions were coming now. So, I mean, it's absolutely true. The major problem of vaccines is that if you don't use them, they don't work. Yeah, that's simple. So the question is how to make them use. Today, especially after the pandemic, they've been politicized. Now there are parties that basically say vaccine is good, the party that say vaccine is bad. So basically that's gone even worse. I think Latin America had actually one champion, Ciro de Quadros, that basically had been able to push vaccination across the continent and be acceptable and be done in high quality. So that's an example. I think the only way to do it is up to us. We need to communicate the importance of vaccines, bring to the policymakers, bring to the people, and communicate honestly and openly the good ways, the risks of the vaccines, and convince everybody that vaccine is an important thing. So it's all about communication, I think. And because we like the science, we like the making vaccines, but we don't spend enough in communicating, education. And I think that's the thing we need to do. For the second thing, I don't think we're going to get soon one shot with every vaccine. But, I mean, we got in pediatrics, we got the hexavalents. Now we have several vaccines for the elderly that are coming up. At some point, we'll need to combine them. So I think combination vaccines will make things easier. But I don't think we'll be able to put everything in one. Unless everything one day we do by RNA, and then looks like, Drew Weissman has put 20 influenza virus into one in mice. Not sure in humans. Yeah.
Thank you very much.
Thank you.